Thank you, Scott. So welcome, everyone. My name is Virginia Chen, and today my colleague Casey and I will be talking about biomarker discovery and challenges and new frontiers facing us in this space. So the goal is by the time you leave this meeting, you'll have a better understanding of the role biomarkers play in healthcare. You'll be aware of some of the pitfalls when it comes to finding biomarkers and ways you can handle these challenges. And lastly, you'll gain familiarity with the new technology that's poised to revolutionize this field. So I'll be covering the first two topics, and Casey will step in to cover the third and the second half of this webinar. So to start this conversation, let's talk about what we mean when we say biomarker and give a little bit of context around their development and use. The word biomarker means different things to different people. So in this webinar, we're going to use the definition shown here. So a biomarker is a measurable indicator of a normal or abnormal biological process or biological response to an exposure or intervention. So for instance, blood glucose is a biomarker, and so are things like heart rate and blood pressure and so on. Now, a biomarker signature consists of multiple molecular biomarkers that taken together form a measurable indicator of this biological process or response. So individual things may not be super informative by themselves, but the combination of them can still be useful. For instance, if we think about how the vital signs are used, they're often used together. So either in an implicit decision tree, or you might count the number of abnormal vital signs when monitoring a patient. So at Proof, we often refer to biomarker signatures as biomarker panels, the way you might have a panel of advisors. So we mean the same thing. Here are some more examples of biomarkers that are used in healthcare today. We see they cover a range of applications and a range of forms. The majority listed here are molecular, so that would be DNA, RNA, protein, or metabolite. There's a few that are imaging-based, and there's also one involving cell culture on this list. Now, the applications range from broad population risk assessment to diagnosis and monitoring of specific individuals with a specific disease. Now, over the past 10 years, the Proof Center has established the end-to-end biomarker development process shown here. So this is a clinician-driven process with careful consideration of healthcare implementation, health economics, and commercialization strategies to ensure user uptake and commercialization success. The first phase is demand analysis, and this involves working with expert clinicians to carefully define what the unmet needs are and whether they can benefit from a new blood test or biomarker. The next phase is developing the content, which in this case means the biomarker itself. So our strategy for identifying a biomarker panel typically involves parallel genomic and proteomic analyses on a carefully selected patient cohort, with analysis performed by technology partners. We then move the most promising biomarker panels into independent replication studies and select those with ideal characteristics based on the intended application for the next phase. The next phase, phase 3, involves working with commercial partners to transfer the biomarker panels onto clinic-ready platforms. So this is the topic of webinar 3 in November, so stay tuned to hear more about that. Using this biomarker development process, the Proof Center has developed biomarker content for 8 major program areas and 14 disease indications since 2009. So now that we've briefly discussed what biomarkers and biomarker panels are and what's involved in going from an unmet need to a clinically useful tool, let's talk about some of the problems and challenges that you might face during the discovery stage specifically. So with limited time and resources, we need to identify the biomarkers with the most potential to be replicated, validated, and translated. So before we talk about potential problems, let's think about what a biomarker discovery pipeline looks like in general. So you start with biological samples, which you then run on your research platform. You take the raw data, you do some sort of processing, quality control, that type of thing, until you end up with a clean data set. This is the type of thing you might be able to open and view in, say, Excel. So we'll call this the processed omics data. Once you have this, you want to come up with a short list of candidate biomarkers that is the most interesting genes, proteins, sequences, etc., as they relate to your question of interest. This can involve many different steps and criteria, but we'll call this list that you end up with your candidate biomarkers. Lastly, you'll probably want to pick just a few of these to build a predictive model around, and this becomes your biomarker panel. So throughout this pipeline, there are many different decision points and things to consider when trying to optimize your biomarker discovery. Today, we'll be talking about a few of these. So, even before we get our process data in hand, we need to think carefully about our analysis design. Sorry about that. And how to think about noise in our data, how to improve the signal to noise ratio, how to deal with outliers and confounding effects. As we try to pick the best biomarker panel to move forward with, we also want to be rigorous in our performance estimation. And as that's one of the criteria that typically is considered when talking about what's the best biomarker panel. So we'll start with batch correction and the implications for analysis design. Now, assuming you've figured out your question of interest, you've gathered samples from your population of interest, one of the next decisions you'll make is how you're going to run your samples on your research platform. So this is often, an afterthought, but you need to put some thought into it to avoid causing issues down the line. So the specific example I'll be showing here is work we did with Megan Levings on a type 1 diabetes studies on blood run on a custom nanostring assay that had 12 samples per cartridge and 6 positive controls per sample. So on the top right, this is a visualization of the positive controls across all of the cartridges, specifically the geometric mean of the positive controls. We can see that there is both a cohort to cohort, that is the different shapes of variation, as well as just random technical variation between the different cartridges, which are the different colors. The points represent individual samples. So this was analyzed over a period of years, and we can see immediately that it wouldn't make any sense to start analyzing our question of interest without first correcting for this kind of variation. So in this particular case, nanostring actually recommends calculating what they call a normalization factor, and then using this to correct the values and readouts from individual samples, as well as using it as a metric to QC the data, maybe throw out some of the samples with too high or too low of a value. So this is what we did in the bottom figure. So we actually did this at the cartridge level because we could see that even within the same cohort, there was variation between the cartridges. So, while normally we might just throw this at a study as a whole, in this case, we decided to do it cartridge by cartridge. And this is the result. So we see that it brings most of the values in line, and there's still a few that fall outside of the range recommended by nanostring, so we removed those samples. Now, the reason this worked in this study, importantly, is because we ran the type 1 diabetes and healthy samples balanced across the cartridges in a roughly proportional manner. So if we had run all of the diabetes samples first and then run all of the healthy samples on later cartridges, there would be no way to correct for the variation we saw without also wiping out some of the signal. So in short, we need to think about our question of interest when designing the analysis of our samples, and then also correct for technical variation after the fact as much as possible. The next pitfall we'll talk about is filtering or pre-filtering. So what is this? It's essentially throwing away some of the analytes you've measured right at the start before even looking at your research question. So why would we want to do this? And the answer is it might actually improve the quality of the results you get. But by throwing away analytes that are noisier, you can improve the signal to noise ratio in your data, which makes it easier to find the signal. It also affects the results of multiple testing correction. So multiple testing correction is when you adjust your p-values for the fact that you're looking at many, many analytes. And so there's a small probability that some of them will look really good just by virtue of how many you're looking at. And it's an important step in looking for biomarkers, but you do get penalized for looking at too many things, especially if there are many noisy analytes in your data set. So the example I'll discuss here is from a type 2 diabetes study looking at gut microbiome. So on the left, we can see that most of the bacterial sequences detected were detected on average fewer than once per sample. So while it's possible that one of these sequences could hold an interesting signal, it's a little bit unlikely. Now on the right, I'm showing the number of differentially expressed sequences at an FDR of 0.05 for more and more stringent levels of pre-filtering. So on the left-hand side, there are 33,000 features without any pre-filtering at all. And then on the right-hand side, we're filtering for about, for sequences that were counted more than about once per sample on average, which leaves us with just shy of 13,000 features. And we can see that in this particular analysis, we actually get more significant results with a more stringent filter, despite the fact that we've reduced our discovery space by over 50%. Now, it's not a rule that filtering out low expressed or low abundance features will always give you better results. It really depends on what you're measuring and the specifics of the platform. So for example, with microarrays, it's well established that there is background noise and you want to correct for that, perhaps filter for that. But you might also want to be careful of genes that are very highly expressed because there are limits of detection, and you'll likely get weaker signal at the top end of the range as well. So there's also other ways to filter your data, for example, based on biology. We published a paper some number of years ago where we used gene expression from heart biopsy tissue to help guide our analysis for blood-based biomarkers of acute rejection. So by limiting our discovery space to genes that were differentially expressed in heart tissue during acute rejection, we were better able to find blood-based biomarkers for the same disease. So in summary, filtering your data in a thoughtful manner can be a useful step when looking for biomarkers in a large discovery space. Another thing to consider is whether or not we have any confounders or covariates that are important to take into account in our analysis. For example, if our initial analysis design wasn't perfectly balanced and we know technical variation is present, we might want to take batch into account at this stage. Or if we know or suspect that age or sex or some other clinical variable affects the analytes we're measuring, or our phenotype of interest, we might also want to take those into account somehow. So here we're going to discuss an example from some work we did with Chen looking at genes whose expression in whole blood was associated with the administration of prednisone. So we knew that prednisone affects cell composition, as seen here both from our study and also in the literature. So naturally, this is going to impact overall gene expression as well. Now, to try and disentangle these effects, we can adjust for cell composition. So here we're showing the p-value distribution without adjusting for cell composition. So we see that there are 1,800 genes differentially expressed at a p less than 1% level. And we know that because of this confounding effect that we're also going to be picking up on genes that are not actually differentially expressed, but rather they're just expressed differently in different immune cells. On the bottom right, we see that after adjusting for cell composition, we identified just 546 genes differentially expressed at the same p-value threshold. So this looks a lot less exciting, but if we're looking specifically for genes whose expressions change with prednisone, and not just genes that are differentially expressed in different cells, we can adjust for cell composition to help us avoid false positives. Our next topic demonstrates why robust statistics are something that should be included in everyone's analysis. Now, this is especially the case when you're doing a lot of analysis, a lot of analyses. This might be the case when you're doing a biomarker discovery type of thing. So you're probably using scripts and programming, and you're not really able to look at all of the results and sift through them in that way. So this example, we're talking about actually COVID-19. And specifically, we were looking at a cohort recruited with Mustafa Toma, and that we were repurposing to use to look for associations between COVID genes and various clinical parameters. So this was work we did with Donson as well. So here's the first of 3 genes that we identified to have a significant association with white blood cells. So first, we can see that the majority of the subjects had white blood cells in a fairly normal range, maybe a little bit high, and then there's one clear outlier. Now, both the Pearson correlation, which is not robust, and the Spearman correlation, which is robust, have nominally significant p-values. So the way we can think about this is, regardless of whether you're ignoring the outlier or including the outlier, you can draw a positive trend line through your points. Now for this next gene, we see a case where the Pearson correlation is significant, but the Spearman correlation is not significant. Visually, we can understand this as the outlier is really driving the trend here, and without it, it doesn't look like the cloud of points has any direction at all. Lastly, we can see the opposite scenario for this gene. So here the Pearson correlation is smaller and less significant than the Spearman correlation. And the way we can think about this is, well, we have an upwards trend in the main body of points, but once you take into account the outlier, the trend line really gets flattened and dragged down. So outliers might suggest a trend where none exists or downplay a trend that actually does exist. Now, the examples here are just simple 2-dimensional scenarios, but I hope they highlight how important it can be to use robust statistics, especially if your data are skewed or you have outliers. Now, almost any kind of statistic you might want to do, there exists a robust variation of. So my last topic is performance estimation. And here we'll be looking at a mass spec proteomic study undertaken with Donson. The first cohort in this study consisted of 72 subjects with paired time points, which we used to find a biomarker panel for COPD exacerbation. Now we want to estimate how well this panel is performing and could be expected to perform in, say, a follow-up replication. Now, for this example, we'll be using AUC as the measure of performance because it's a kind of general measure that doesn't require us to know things like disease prevalence or cost of misclassification or things like that. Now, to illustrate what the AUC is for those that aren't familiar with it, here's a hypothetical scenario where we have some healthy subjects, some disease subjects, and some biomarker test that gives us a score as shown on the y-axis. We can see that the healthy subjects tend to have higher scores compared to the disease subject. But if we were a doctor, how do we actually decide, okay, this person is healthy, this person has a disease? Well, we have to draw a line somewhere. Now, depending on where we draw this line, we'll get some percentage of the healthy subjects correct and some percentage of the disease subjects correct. And this is sensitivity and specificity. Now, whatever these percentages are, we can take those numbers and we can draw them on another figure. Now, if we keep repeating this exercise and drawing the line at different points and recording the resulting sensitivity and specificity, we will get a curve. And the area under this curve is the AUC. Now, an AUC of 1 corresponds to a perfect classifier where you can draw a line and all of the points on one side are healthy and all of the points on the other side are disease. An AUC of 0.5 corresponds to a coin toss. And so we can think of 0.7 to 0.8 as being a decent classifier. So now that we introduced this performance metric, let's move on to the example itself. Now, a fairly common way we see in published papers is that people will take all of their training data and for the training and then use all of that data as test as well. So this results in an optimistic estimate because it's circular and your biomarker panel is biased to work well in your discovery samples. Now, using this method for this particular data set, we get an AUC of 0.75 for the study. Now, if your study cohort is large enough, which is unfortunately not the case in many molecular studies, you can divide it into a training and test set. Now, depending on how rigorous things are, you might start to wonder, well, did we pick the right test split? Did we pick the right set of samples to use this test? Should we maybe try this a couple times and pick the best? Should we try this a couple times and take the average? So it does raise a lot of questions. Now, here just as an illustration, we took the data set, we randomly divided it with an 80-20 split, and we did this 10 times to get a sense of the range of AUCs we could expect. And we can see that it's actually a very wide range because it really depends on just randomly which samples were selected to be part of the test. So, this is why questions come up as to, did we pick the right split? Now, the average here is 0.71, so not too far from the AUC that we saw in the first example. But related to this concept is what's known as cross-validation, which is a more systematic and rigorous way of conducting the training test split multiple times. And it's done in such a way that every sample gets to be part of the test set exactly once. When it comes to getting a fair estimate of performance, it's also really important to distinguish between what we call shallow cross-validation versus deep cross-validation. So something we've also seen is that people will find biomarkers using all of their samples, and then they apply cross-validation to the very last step in the process, which is model fitting. But the problem with doing it this way is that when you're doing differential expression and biomarker selection, you're actually incorporating a lot of information from your training data. So if you're using 100% of your training samples, you're actually biasing your results. Now, when we do that with this data set, we get an AUC of 0.75, which is exactly the same as when we trained and tested in the same samples. In deep cross-validation, we would do as many of the biomarker discovery steps as possible within the cross-validation itself. And so the 80% that is being used for training is a separate set of samples from the 20% that's used to test. Now, typically, we might do things like batch correction outside of this and use all of the data because that doesn't involve phenotypic information, just technical metadata. Filtering can go either way depending on the type of filtering and whether you're using phenotypes implicitly or explicitly. And everything else would be done within this train test split within the cross-validation. So in this example, we get an AUC of 0.73, which is lower than what we get from the shallow cross-validation. In this example, there's not actually a huge difference between the less biased and the more biased methods. And this is because we had 72 subjects in each group, and our biomarker panel was actually only 5 proteins. The amount of bias really depends on the size of your discovery space, how large your sample size is, how many biomarkers are in your panel, and also the specifics of the classifier used. But in general, it's best practice to ensure that your test performance is based on samples that were not used for any sort of biomarker selection, biomarker training. So to recap, here are some of the challenges you might come across when doing biomarker discovery and how you can address them. So there's a lot of things we haven't covered today, such as how to pick biomarkers that have a reasonable chance to survive translation onto a different platform, or choosing the right performance metric and the right subset of samples to calculate performance in based on the intended use of your biomarker panel, or designing cross-validation when you have paired samples or you want to balance by phenotype or other clinical variables. But hopefully you learned something new today that can help you in whatever research you're doing. So with that, I'll hand things off to Casey to talk about single-cell technology and how that's changing what biomarkers might look like in the future.
Thanks, Virginia. Bear with me as I try to fight my way through Zoom screen sharing. Okay, can anybody just thumbs up me if you see this? Thumbs up? Yes? Yes, good, okay. So without further ado, I'll dive right in and give you a brief overview of how we're thinking about biomarker discovery using these new single cell assay technologies. So this is going to be more of not so much a lessons learned because the technologies are very new, more of things you need to consider when you're doing this type of analysis in the biomarker context. So we're briefly going to talk about motivation, why you might want to conduct a single cell experiment. We'll talk about two of the major differences when it comes to biomarker discovery, differential expression analysis that are specific to single cells. Or single cell assays, namely dimensionality reduction and clustering. And then finally, we'll have a brief discussion about what a single cell biomarker might look like and what the advantages might be of a biomarker identified using these technologies. And so on the right-hand side of this slide here, I've got something to keep in mind, a useful sort of mental model for the type of technologies we're discussing in this latter part of the talk compared to what we've been discussing so far and what's been sort of prevalent historically in the omics space. So if you're studying complex tissue, which I'm representing here as a fruit bowl, this is shamelessly lifted off Twitter, not my invention, but you're studying this complex tissue, it's a fruit bowl. In the past with bulk omics, you would have done an analysis that would have effectively blended all of those fruit together. And then you might have been able to compare the color of the resulting smoothie in, say, healthy and disease subject populations. And that's informative, right? The color of the smoothie might tell you, oh, someone's sick, someone's healthy. But that's about, in most cases, all the information you would have. So you wouldn't necessarily be able to do very much apart from the diagnostic aspect because you wouldn't know where the change happened, what cell population, and mechanism might be difficult to reason about with that kind of output. So you would do a lot of additional downstream analysis. You had the ability to do a bit more complicated, shown in the middle here. Maybe you did some flow sorting to look at specific cell populations with the same bulk technologies. That would give you a bit more information, but is still not ideal. It's still kind of an average picture over a bunch of cells. And now you can do what we're showing here on the bottom, which is with single cell assays, you can actually study the individual fruit within this sort of fruit salad model of your tissue and finally appreciate the full complexity of what's happening in health and disease, which is quite neat. And so, well, why might you want to do that? Here I've highlighted like three toy examples of the types of changes that can happen in a tissue that is under some sort of perturbation. So the first change that you might observe when applying a perturbation or control or comparing health and disease is a change in the abundance of underlying cell populations. So I'm showing here on the right a tissue made up, really simple tissue, made up of two cells, red cells, yellow cells, who express red genes or yellow genes. And in healthy subjects, we've got only red cells. Yellow cells aren't really doing much of anything. And then, or there are a few of them rather. And then in disease, we have more yellow cells in addition to the yellow cells. And so at the gene expression level, we might detect more of the yellow gene now. And if we were doing a bulk assay, what we would observe is simply, red in health and orange, a combination of the two cells in disease. A different kind of change that could also occur is a change in the expression of the underlying cell populations. So in the second example here, I've got both red cells and yellow cells in the same amount in my healthy tissue on the left. But in my disease condition, the yellow cells are now expressing more of their yellow gene, and that's shown here with a taller yellow bar. And again, if we were just using a bulk assay technology, we would notice the same exact pattern, red versus orange in health versus disease. And then finally, a different type of pattern that might occur is what I'm calling the appearance of a novel cell type. So this is where my analogy breaks apart a little bit, but bear with me. We've got the same situation with mostly red cells in our healthy tissues, very few of the yellow cells. And then rather than getting an influx of yellow cells or an overexpression of the yellow gene in yellow cells, instead what happens is the red cells modify their behavior and express the yellow gene now. And again, here, if we were just using a bulk assay, we would notice exactly the same pattern of red versus orange. And so that's been a fundamental limitation of bulk analysis. We observe differences, but it's very difficult to ascribe them to particular mechanisms. And so they're useful diagnostic biomarkers, but not necessarily actionable, and they don't immediately suggest mechanism of action or potential treatments. So next I wanted to briefly discuss what the analysis of single cell data looks like at a really high level and contrast it to the bulk analysis that we've been discussing so far. So you start with the same kind of data matrix, right? So we've got genes in the rows and samples or cells, depending, stored in the columns. You might optionally collect some metadata information on either the genes or the cells. So this could be things like the age of your samples or whether a gene was a housekeeper or not. And you can store those alongside the primary data matrix for filtering purposes, for example. And then you would go through a number of steps as part of your pre-processing that Virginia talked about a little bit earlier. So things like QA/QC sort of things, normalization. It's very common to do various transformations on the data, and then pre-filtering, which Virginia also discussed. And all of those steps produce transformed versions of the data, which you can also store alongside. And that basically is where you would start your bulk analysis, right? You're done. You've done your pre-processing and you're ready to ask questions of the data. But in the single cell assay space, you've got a little bit more work to do before you're ready to answer questions. And those come in the form of two additional steps. The first is dimensionality reduction. I think so. The most commonly known approach is probably principal component analysis, or PCA, which of course does come up sometimes for bulk analysis, but it's typically just done for visualization purposes. In contrast, in single cell space, it takes a much more important role in the analytical pipeline. And additionally, you might also have different methods of dimensionality reduction, which will come up. So some of you that may have been dealing with this type of data already would have heard of things like TSNE or UMAP. Once you've done the dimensionality reduction step, you can store those alongside your primary data matrix. And then you would do one or more clustering on those lower dimensional representations of your data to obtain sort of putative, I'm gonna say putative cell clusters. But basically assign the samples in your experiment to groups which may represent cells. And once you've done that, and added that data to your metadata table, you're able to interrogate those cell clusters and start to ask questions like differential expression, but also pass that on to biomarker discovery exercise. So next we'll discuss those two different steps in a bit more detail. So dimensionality reduction, I've said the words a couple of times, but I haven't really properly defined it, so we'll start there. At a really high level, it describes a class of methods that aim to reduce the number of variables that you have to contend with in your data. And I'm showing this pictorially here by showing you this toy example of a heat map of a very small primary data matrix with 4 cells here and 17 genes in the columns. And if I were to apply principal component analysis to reduce the dimension of this data, I would end up with something like what I'm showing on the right. And effectively what's happening is that you can notice that we've got this group of genes in the top here that's highly expressed in all of my cells, and we have a different group of genes at the bottom that is relatively lowly expressed in all of my cells. And those two big patterns of the data are being captured now by two components, PC1 and PC2, which account for most of the variance that existed in the primary data matrix, 67 and 29% respectively, and have allowed me to go from 17 different variables to consider down to 2. So why might you want to do that? Well, I think the first thing relates to something that Virginia had mentioned earlier, which is robustness. So while there may be measurement error in some of the individual gene measurements, if we consider aggregates, averages of many genes that are correlated, then that measure is a bit more robust to that measurement error effect. But a second and maybe a more important reason to do this in the context of single cell data is a reduction in the complexity of the data. So we went from 17 variables to 2. And that's really important because some of the downstream steps, so the next step, for example, clustering, can be very computationally expensive when dealing with large data matrices. So I'm showing an example here on the left that is the result of clustering on the input data using an increasing number of variables. So I've got 4,000 cells in my data, and if I use fewer than 100 variables, I can do the clustering in under a minute. But if I go up to 50,000 variables here, it's going to take me close to 3 hours, which obviously, if you're doing it once, it might be okay. But if you're doing it a number of times using different methods, that might be become a little bit more taxing. The other side of this is the memory requirements, which typically also scale the same way. And so it might be the difference between running your data on a laptop and having to go to a server. Once you've got your lower dimensional representation, you're going to do some clustering, and that can be, so you could do dimensionality reduction in a couple of ways. I mentioned PCA already. There's also t-SNE and UMAP. And really, I don't have time to go into detail here. What I really wanted to show is simply that the representation is different, so you'll get different results or different representations depending on the method that's applied. And really, the more exotic or the newer methods like t-SNE and UMAP were mostly applied to single cell data because they give you these tighter groups, which may be easier to cluster. But they come with a lot of downsides too. They're computationally more taxing to compute, and they're also significantly less interpretable. And if you've got questions about that, I'm happy to discuss them at length. Please email me. But once you've got your lower dimensional representation, whatever tool or approach you use to compute it, you'll cluster it. And that is shown pictorially here by going from these gray spots on the left to some colored spots on the right, which correspond to an assignment of the individual dot or the cell in this experiment to a group of cells, which can now be considered to be similar and potentially represent a distinct cell population in the data. And again, this you can do a number of different ways. They all have their pros and cons. All of them have parameters that you can explore. But I think it's important to remember that whichever method you apply and whatever clustering you obtain, it's still only a hypothesis about the identity of the cells within that cluster. And so it's important to think about validating that cluster assignment. And you can do that a number of ways, sort of calling back to what Virginia was talking about when she discussed cross-validation. You can do this in a data-driven way by doing some bootstrapping or cross-validation. And I included a citation here where we did something very similar when identifying correlated gene modules a few years ago. But you could also think about validating using biological information about the tissue that you're studying and leveraging the literature. So considering the clusters, the genes within that define those clusters and comparing them to cell-specific signatures that you know about and seeing whether they line up properly. Once you've defined those clusters more than once, probably a number of different approaches, and validated them or are confident in them, you can start asking questions of them. And that looks like this. So we're doing differential gene expression, and we noticed that cluster 4 is expressing this gene more highly. But you can also do biomarker work. So if you had cells from many patients within this experiment, you might notice that your cluster 4 actually neatly segregates your patients with your healthy patients in blue from your diseased patients in pink. And so that could be quite exciting potentially. And the reason I say that is because of this kind of thing. So this is my motivating example for being excited about single cell biomarkers. This is from a study on the right-hand side, a figure from a study recently published in Nature. And what it's showing is the survival time in various samples of breast cancer tumors. So the mean survival for this particular disease is in black. It's roughly 60%, and that's analogous to the smoothie, right? So that's what we know right now. And what these researchers were able to show was that by qualifying the expression profiles in a particular cell population, the basal CK- cells, they were able to identify two different subgroups of those of the patient population. One subgroup had basal CK- cells that had this apoptotic phenotype, and they had 100% survival in the study. And that's being shown here with the nice sort of very fresh looking yellow fruit in the right. And then conversely, they had a subset of the subjects that had basal CK- cells that had hypoxic phenotype. And those subjects only had a 40% survival, which is obviously far worse. And so the really nice thing is that in one study, you've immediately identified a potential mechanism to explain this difference in the survival rates of these patients. And you can immediately start looking at therapeutics without any follow-up work, which is tremendously exciting. And so in summary, single cell assays, new and exciting, yes, but significant computational costs. And if you run up against some hurdles, please come talk to us. We're keen to do more work in this space. The analytics are evolving, and I hope I've convinced you with my overview that they're at least an order of magnitude more complex than for bulk. But nevertheless, super promising stuff for biomarker work because of the high resolution, interpretable within one experiment, and then potentially actionable, immediately actionable, which is very cool. And so with that, I'd like to thank everyone for their attention and plug the next webinar, which is going to be on October 16th. Dr. Armit Singh and Daniel He with Proof are going to be talking a little bit more about multi-omic data. And I guess if we have time, we'll take some questions now. Thank you.